Good afternoon.
Right. Last class is the cell organelles. Cell organelles is sixth chapter. The cell structure and functions is over. Right. Amino acids. Right. Cell structure and function. Sixth now discuss in the sixth unit. Right. All of you once again. Please respond. Cell organelles topic. Cell organelles. Right. Is it visible?
Yes, sir.
Right. Cell structure and functions. The prokaryotic cells is the types of cells. For example, this structure first basically. So cell was first discovered by Robert Cook. Is it visible?
Yes, sir.
Yes, sir.
Cell was first discovered by Robert Hooke in 1665. Yes. What is the cell? Structural functional unit of the life. Yes. Cell is the structural functional
Function of the basic unit molecule.
Okay. Next. These are prokaryotic cell, right? These are prokaryotic cell and ribosomes. This particular part of the ribosomes, and these are flagella. These are to the DNA, nucleated DNA. What is the DNA? Deoxyribonucleic acid.
Deoxyribonucleic acid.
Prokaryotics have to the only for nectar DNA, no nuclei. There is no nucleus. Few internal structures, right? One-celled organism, bacteria. Ribosomes. Ribosomes are 70S ribosomes, 35S ribosomes. Right? This particular layer of the cell wall. These are cell membrane. These are cell membrane. like
Sir, disturbance వస్తుంది.
right, structure of the cell, this particular structure of the cell. These are plasma membrane. These are plasma membrane. These are nucleus. This particular part of the rough endoplasmic reticulum. Rough endoplasmic reticulum. And mitochondria. These are mitochondria. Ribosomes. Lysosomes. Vesicles. Cytoskeleton. Peroxisome. Structure of the cell. Right? Nucleus is the coordination of internal cell organelles. Right? Structure of the cells through nucleus is the chromatin. Nucleopores are there. Nucleopore envelope. Nucleus. And glycogen, rough endoplasmic reticulum, ribosomes, Golgi complex. These are to the Golgi complex, right? These are lysosomes. White, is it visible? These are lysosomes. These plasma membrane. These are plasma membrane. Centrioles, right? Mitochondria is the powerhouse of the cell. Mitochondria is the powerhouse of the cell. This particular part of the mitochondria, these are all cell organelles. These are all cell organelles. Cell organelles. These are all cell organelles. In the particular, in the specification of in the centromeres and centrioles, plasma membrane, lysosomes, smooth endoplasmic reticulum, rough endoplasmic reticulum. These are to the rough endoplasmic reticulum. These are smooth endoplasmic reticulum. Rough endoplasmic reticulum, this particular writing is there. Ribosomes. Ribosomes are the 70S ribosomes, 80S ribosomes, right? Microfilament. Covered with these are to the microfilament. Isolation of submolecular organelles, homogenization, homogeneous cells described by in the isomatic. These are to the isolation of subcellular organelles. Isolation of subcellular organelles. Parts of the cell: plasma membrane, nucleus, and cytoplasm. Right. Now discuss nucleus. Nucleus is the prominent of large organelles. Nucleus is the prominent of large organelles. Houses of most of cells of DNA. These are most of cells DNA, right? Contain genes as well as in the controls of the activity of other organelles. Activity of other controls the activity of other organelles. The coordination of the all cell organelles. The contain genes. The houses most of the cells of DNA, right? These are nucleus. Nucleus structures means in the spherical or oval shaped structure. Nucleus is the spherical or oval structure. These are oval structure or spherical structure. Next, as well as the the most cell have single nucleus. All right. Single nucleus means, for example, these are single nucleus to the RBC, nature of RBCs have none. Contracts the skeletal muscle cells have multiple nuclear envelope covered with the nuclear envelope. These are covered with the nuclear envelope, right? The multiple nuclei as nuclear pores. Each nuclear pore consists of circular arrangement of the proteins surrounding central opening, right? For example, these are to the nuclear pores are there, number of nuclear pores. These are to the number of nuclear pores to the as well as to nuclear pores and consist of circular arrangement of the proteins surrounding of the large part. Thus, the chromatin layers are there. Main function controls cellular structures, distraction of the in the directs in the cellular activities, produces of ribosomes in nuclei. Produces of ribosomes in nuclei, nucleopore, nuclear envelope, details of the nucleus. These are layer of the chromatin. These are rough endoplasmic reticulum. These are rough endoplasmic reticulum. Ribosomes. Ribosomes are the sites of protein synthesis. Ribosomes are sets of protein synthesis. The name these are to the thinly organelles. Reflect there in the high content of rRNA. They are either attached to the filament in the 3D cytoplasm. Consists of two subunits are there, large amount of, right? Large subunits, small changes of the ribosomes to the in the protein synthesis. Ribosomes are the mainly protein synthesis, right? Complete functional ribosomes, large amount of changes to the ribosomes. And endoplasmic reticulum. These are particular structure, endoplasmic reticulum. Endoplasmic reticulum is mainly two types. A network of membrane in the form of plaited of sacs or tubules, like that structure of the in the internal cell. Two type, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Two types are there, endoplasmic reticulum. Network of membranes. Endoplasmic reticulum means the network of membranes. The formation plotted as the sacs tubules as like that to formation of the internal cell organelles. Right? Structure of endoplasmic reticulum. Look here, all of you. Is it visible? Nuclear envelope. These are red shape like that structure, nuclear envelope. These are ribosomes. The smooth ER means smooth endoplasmic ribosomes. These are to the rough endoplasmic ribosomes. Right? The ribosomes means network of membrane. What is the functions of ribosomes? Network of membranes from the plotted sacs of the tubules are there. Right? The ribosomes, particular network of membranes. Functions of the endoplasmic reticulum. Rough endoplasmic reticulum means only for rough endoplasmic reticulum synthesis proteins. Synthesis of proteins. And post-translation modifications only, such as plastification, as well as the phospholipid, cholesterol, steroid hormones, adrenal glands, and synthesizes to the phospholipids, cholesterol. Many tissue of this, these are to the in the phospholipids, cholesterol, steroid hormones, right? Glycogen metabolism, stories in the releases of calcium ions in the cells, right? Thus, detoxes in the drugs, harmful substances, removes the phosphate groups in the G6 and P, release free glucose in the blood. Golgi complex. These are discovered the Golgi complex. It consists of 3 to 20 small flatted membrane sacs. Small flatted membranous sacs. Most cells have the Golgi complexes, right? Golgi complex are more extensive cells that secrete proteins, okay? Right, they like that structure modified to the transporters of proteins. What is the functions of transportation of proteins? Received from the rough endoplasmic reticulum, Golgi complex. Right, these are to the proteins by the Golgi complex. Right, ribosomes, synthesized proteins. Right, rough endoplasmic reticulum, smooth endoplasmic reticulum, transverse vesicles. Right, entry phase of the cisterns, medical cisterns to secretory vesicles, processing and packaging of proteins by the Golgi complex. Mitochondria, next organelles of the one of the mitochondria. Mitochondria is the powerhouse of the cell. What is the function of the mitochondria? Powerhouse of the cell. Elongated, round shaped, elongated, round these are to the mitochondria. Mitochondria means powerhouse of the cell. Active cells such as muscles and as well as the active cells such as muscles, liver and kidneys which use ATP at high rate. What is the ATP? Adenosine triphosphate. What is the ATP? Adenosine triphosphate, right? Adenosine triphosphate. Adenosine triphosphate. These are energy currency of the cell. These are energy currency of the cell. These are energy currency of the cell. Energy currency of the cell. Production of energy as well as these in the produces to cell organelles in the powerhouse of the cell, hundreds as many several thousands of the cell organelles. These are to the mitochondria, inner mitochondrial membrane. These are to the inner mitochondrial membrane. Matrix. These are matrix and these are cristae. Ribosomes. These are ribosomes. Enzymes. These are enzymes. Outer mitochondrial membrane. These are outer mitochondrial membrane. Inner mitochondrial membrane. Inner mitochondrial membrane. Generate ATP through reactions of the ATP, adenosine. Ribosomes. Ribosomes, enzymes, outer mitochondrial membrane, inner mitochondrial membrane. These are to the outer mitochondrial membrane. These are inner mitochondrial membrane. Matrix. Cristae. Lysosomes. Lysosomes are enclosed by vesicles from Golgi complex. 60 kind of powerful digestive hydrolytic enzymes. Right? Lysosomal enzymes also helps recycle round out cell structure. Right? Some disorders are caused by in the absence of lysosomal, example, Niemann-Pick disease and change to lysosomal. This like that structure. Lysosomes, okay. These are to the membrane enclosed of vesicles are there. That from the Golgi complex. Golgi complex, powerful digestive and hydrolytic enzymes. Digestive and hydrolytic enzymes. Lysosomal enzymes also help and as well as a recycle out cell structure, helps in fertilization. Some disorders are caused by in the absence of lysosomal enzyme. Chromosomes means smaller structure of lysosomes. Smaller size contains several oxidases, enzymes that can oxidize various organic substances. Right? Degradation of amino acids, oxidation of long chain fatty acids, oxidize toxic substances such as alcohol to changes. cytoskeleton. Cytoskeleton network of protein filaments extend through cytoplasm. Filament like structures, tubules. These are to the cytoskeleton. Microtubules, unbranched polar cycloids, made up of protein, tubulin, the largest of cytoskeleton components. Right? These are microfilament. Microfilament means elements of cytoskeleton composed of the protein chain, action, intracellular protein that just under the plasma membrane, most prevalent edge of the cell, as functions mechanical support for the basic strength shapes of the cells. Microfilaments are involved in muscle contraction, cell division, cell locomotion. These are to the cytoskeleton. Right? Cell organelles to this particular topic, the metabolic and DNA biomolecule chromonucleus to biomolecules. Organ units in the carbon chemical evolution of DNA. These are topic is the compression of plant cell and animal cells is over. Organelles and other functions, cell organelles. Structure of prokaryotic and eukaryotic cells. The 60 unit is over. The biology of students without biology in this. Alright, block 2, 60 unit is the particular topic is over. Next topic to and 9th unit 9. The structural organization of proteins. Right, these are to the amino acids and peptides and proteins. These are 9th chapter. Enzyme, cell cycle, cell division, and onion root tips. Eukaryotic cell cycle, mitosis process, chromosomes. Right, amino acids and peptides and proteins. Now topic. Amino acids Amino acids. Amino acids, proteins. Right. Amino acids and proteins. Right. Amino acids and proteins. These are to the each strand of DNA holds to create proteins. Proteins to particular the because of DNA leaves the copy nucleus cell. Copy of the code called as messenger RNA, mRNA is made. These are called as transcription, right? The mRNA takes in this information. Messenger RNA outside the nucleus being to the ribosome, that is called as messenger RNA. The one information, one place to another place, translation or transcription, that is called as to messenger RNA. Messenger RNA means take this information outside the nucleus or the brings into the ribosomes, right? As well as the inside the nucleus, inside the cell wall. These are to the amino acids. These are to the mRNA. What is the function of the mRNA? Take place to the information outside the nucleus, brings it to the ribosomes. Ribosomes move along the mRNA reading according in the phase of translation. Translations means one information. Picture is not clear. Okay na? Message వస్తుంది not clear. Okay na ఇప్పుడు? Is it visible? Right? Okay na?
Okay, sir.
Right. These are to the ribosomes moves along the mRNA. The code of this the phase of called as translation. Ribosomes move along the mRNA reading the code. This is the phase of called translation. What is the translation means? The messenger from to the ribosomes along the mRNA reading, that is called as translation. Another type of RNA called transfer RNA means tRNA. Like that, these are to this particular tRNA. These are tRNA. tRNA collects the specific amino acids that needed to the make of the position to the proteins are difficult to the different types of 20 different tRNAs, each one of the amino acids. tRNA brings the amino acid to the ribosome, right? Brings the amino acid to the ribosomes. Ribosomes then bonds, then chain of amino acids, the protein in the proper sequence based on the code in the mRNA called elongation. mRNA means the ribosomes continues to downward to the mRNA strands until approximate amino acids added to the protein in the complete division to the specification of mRNA. Messenger RNA, transfer RNA. What is the function of the messenger RNA? Only for leading code of this in the phase of called translation. Translation is called as messenger RNA. tRNA means these type of RNA to in the specification of amino acids to needed to the protein. These are based on the ribosome, right? Amino acids, the essential of the proteins is the essential of the bodybuilders as proteins as converted to the amino acids. Amino acids means particular, these are to the process of change to one internal of the body organelles to proteins and amino acids in the bodybuilders to growing of physiological activity. The physiological activity of the human beings. These are to the in the specification of proteins. Proteins means large molecules made up of chain of amino acids. Proteins as a made up of amino acids. What is the amino acids? Like that proteins, bodybuilders. For example, development of the body growth, muscle growth to protein food materials. Like that, the chain of the amino acids to proteins. Proteins is converted to amino acids. Every cell in the body or most of the body functions life process to the proteins. The sequence of amino acids is determined by the DNA. The sequence of amino acids is determined by DNA. What is the DNA? Deoxyribonucleic acid. DNA means
deoxyribonucleic
acid. Okay. Proteosis means holding the first place. The chemist suggested by the molecular living system, molecule abdominant of the molecular liver system, cellular derived. Friedrich Sanger won Nobel Prize for achievement of 1950 insulin. These are structural differences between carbohydrates, lipids, and proteins. Carbohydrates in the chain of glucose. Carbohydrates, chain of glucose. For example, what is the first of all the basic function of the what is the digestion? Complex molecules are converted to simple molecules is called digestion. Yes or no? Complex molecule, for example, taking of macronutrients, carbohydrates, lipids, and protein. Carbohydrate digestion means taking of food material carbohydrates, complex molecules are converted to simple molecules. That is called as digestion. Digestion is the mainly two types are there: intracellular digestion, extracellular digestion. Intracellular digestion, within the cell in the, for example, the microorganisms like that bacteria or virus in the intracellular digestion. And extracellular digestion means development of human beings, development of mammals. That is called as extracellular digestion. Carbohydrates to the polysaccharides converted to the disaccharides. Disaccharides converted to the monosaccharides, which is the enzyme activity, maltase enzyme. Carbohydrates, these are in the process of maltose enzyme, glucose. Carbohydrates converted to the glucose units. Lipids converted to the digestion fatty acids. Triglyceraldehyde, fatty acids. Look here in the slide. In the carbohydrates converted to polysaccharides and monosaccharides and disaccharides. Disaccharides and monosaccharides. Lipids as a conversion of the digestion to the fatty acids as triglycerides. These are the triglycerides. Proteins as converted to the amino acids. Okay, now previously one student is asking the what is the function of proteins means. Proteins is conversion of the chain amino acids. Okay, now who is the asking of question ever ante ఒకసారి చూస్తా నెంబర్ లేదు కానీ importance of amino acids to the in the body builders of the proteins of the human body, right? Vijay, Ravuri Vijay abbayam Ravuri Vijay. Ravuri Vijay is a protein has converted to and the amino acid chain. These are to the amino acid chain, right? Amino acids, amino group containing carboxylic acid group, right? Amino acid has a central carbon atoms with 4 different groups of atoms. Amino acid, NH2 group and CO, carboxylic acid group means COOH group. Hydrogen atom, radical CH3 and CH2. Nomenclature. Classification of amino acid based on structure and spread chains to the amino acid acidic mono amino dicarboxylic acid and basic amino diamino mono carboxylic acid. These are to the polar amino acids, non-polar amino acids. Polar amino acids means hydrophilic chain without charge, acidic nature, negative charge, basic, and that is called as polar. Non-polar means hydrophobic slide chain of the no change, right? That is called as to non-polars. Ninhydrin, hydrogo, expellent, amidation reaction due to the NH2 group. Functions of amino acids. Act building block of peptides, polypeptides, and proteins. Okay, now amino acids act as a building block of peptides, polypeptides, and proteins, right? Abnormality and peptides and polypeptides and proteins. These are to the respectively right. Glycogenesis, glyconeogenesis means glucogen is converted to the glycogens to participate of synthesis of specialized products, neurotransmitters. What is the neurotransmitters? Hormones as a neurotransmitter. What is the hormone? Hormones as a chemical messengers. What is hormones as a chemical messengers? What is the specific of chemical messengers? These are hormones as to, for example, releasing of the pituitary. How many types of hormones are there? Two types of adrenohypophysis and neurohypophysis. Adrenohypophysis releasing of the hormones FSH, LH, and MSH, as well as the TSH, to follicular stimulating hormone, luteinizing hormone, melanocyte stimulating hormone. These are to the prolactin hormone. All type of hormone releasing of the peptide hormones only for anterior pituitary. These are to the hormones means like that hormones, neurotransmitters like that hormones, testosterone and estrogen. Covalent bond means sharing of electron pairs, that is called as covalent bond. The double bond character to change. These are to the catalytic proteins, catalysis process enzymes. Example, structural proteins support the framework of cell, collagen proteins and elastin. Transport proteins, transports of substances, albumin and transferrin. Hormonal proteins means regulation of function, insulin and glucagon. Gene regulatory protein like that regulate of genetic function, histone and chromatin. And protective proteins means prevent function, immunoglobulin. Receptor protein, receptor function, LDL receptors. Contractile protein, muscle contraction, actin and myosin. Right. Mari proteins as a building blocks and endukannamanante, now how to estimate in the protein blocks of building ante every function to the in the body, the framework of cells. For example, you suffering from fever, you suffering from disease, how to in the eligible of how to in the protection of the body, taking of food materials, taking of immune regulations of the food materials. That is the proteins. You have to in the internal body proteins. As functional and classification types of proteins, structural proteins are there. Catalytic proteins, transport proteins. For example, transport of proteins substances, albumin and transferrin. Albumin is a transportation, one place to another place in the body. Injuries of the internal of in the cell organelles to transport of the protein regulations. As for example, hormonal of the protein. Insulin, one of the regulation of the function to the secretion of the insulin to in the endocrine glands. Glucagon, right? In the gene regulatory protein, regulate genetic functions, histone and chromatin. The histone proteins are there and chromatins. Once again, look here. Functional classification of proteins, structural proteins, catalytic proteins, transport proteins, hormonal proteins, gene regulatory proteins, protective proteins, receptor proteins, contractile proteins. Structural proteins means framework of cells, collagen, elastin. Catalysis proteins means enzymes. Catalytic proteins are enzymes, trypsin, chymotrypsin, lipase, amylase. These are to the enzymes, trypsin, chymotrypsin, lipase, amylase. This type of to the enzymes, right? The transport of substances means albumin or transferrin. These are to the transportation of the proteins. Hormonal proteins. Hormonal means regulation of the hormone function. For example, pancreas and as well as the endocrine glands are exocrine glands. Pancreas is the endocrine gland and exocrine gland. That is called as compound gland of the endocrine system. Compound gland of the endocrine system. Compound gland of the endocrine system means simultaneously two functions are there. Simultaneously two functions and means there is a specification of the two functions means endocrine function and exocrine function. That is the functions of the insulin factors to the in the protein, hormonal proteins, right? As well as the protective proteins prevent infection. Prevent infection, protective proteins. Protective proteins. The prevention of immunoglobulin. Immunoglobulin means innate immunity and acquired immunity. What is the innate immunity? Transmission of heredital characters. For example, ancestors of the family. మీ యొక్క తాత తండ్రుల నుంచి వచ్చేటువంటి these are to the in the parents to grandparents, The transmission of herediitary characters, one generation to another generation, these in the ancestors of the family. That is called as to the specification of immunoglobulins. And immune system regulations is the mainly two types are there. One is the innate immunity. Second one is the acquired immunity. Right? Innate immunity and acquired immunity. Those are in the ancestors of the family, the transmission of one generation to another generation, in the classified and the change of characters, in the change of genetical disorders, in the one transmission of prevention of the prevent infections. That is called as immunoglobulin. The proteins act in the functions of the immunoglobulin. Receptor proteins means LDL receptors, chemoreceptors, thermoreceptors, mechanoreceptors. These are act as a functions to the proteins, right? Mechanoreceptors and electronoreceptors. These are combination of the electron. These are process of the chemical composition of chemoreceptors. Electronic transduction of the electronic molecules, that is called as electronoreceptors, and chemoreceptors, thermoreceptors, that in the receptor proteins are there. And contractile protein. Contractile proteins means muscle contraction. Muscle contraction means actin and myosin. Actin and myosin, these are muscle contraction, right? The actin and myosin. Simple proteins means without any non-protein substances. That is called as simple proteins. Globular proteins, albumin and globulin, histones. Spherical shape, water soluble, digestible, oval shape, globular proteins, albumin and globulin, histone proteins. Conjugated proteins, composed of simple protein along with non-protein prosthetic substances, nucleoprotein, nucleohistones, nucleic acid, HDL, lipids, glycoproteins and carbohydrates, mucin and carbohydrates, chromatoproteins. Chromatoproteins means cytochrome, heme, hemoglobin, meta operate in the carbonic changes. Delivered proteins means protein, metaprotein, peptone, peptides, amino acids. Peptones, peptides, and amino acids. Peptones and peptides and amino acids. As well as the secondary derived proteins as the produced by hydrochloric cleavage bonds, peptides. The peptide proteins like that, amino peptide proteins, the functions of the peptide amino acids to change of character. Peptones, monopeptide, dipeptide, polypeptides, formation of the monopeptide, dipeptides and polypeptide chains. So the peptide proteins. peptide proteins. Is the peptide amino peptones, peptidases, amino acids, and as well as the secondary derivatives of the proteins, right? Classification of proteins complete. Best sources of proteins: daily fruit, dairy, meats and poultry and meats, food. Properties of proteins: pH of the sequence solution, which has molecules, average have to net change, structure of protein levels, primary structures, secondary structures, these are to the secondary structure, tertiary structure, quaternary structure. Helix, these are amino acid residues, helix, polypeptide chains. Secondary structure, most common suitable conformation, helix. Parallel top view, tertiary structures, myoglobin. These are myoglobin. Quantity structure, quaternary structure, alpha chain, beta chain, affecting of protein structure, hydrogen bond, electrostatic interaction, hydrophobic interaction, then a string of agents, heat, organic solvents, mechanical mixing strong, flexible structure framework of the cells and system, cytoskeleton, provide and mechanical structural proteins, defense of ethnic function, antibody, muscle contraction, source of energy, acts as buffer, promote catalytic function, hormonal function, genetic information state storage, expression, transmission, right? These are to the in the proteins as amino acids, the chapter 6th chapter, right? Proteins and amino acids is over in this session, right? Amino acids to these are to the amino acid functional organic molecule contain both carboxylic group, COHS group, as well as the amino group. NH2, the amino acids derived from proteins, have amino group on the alpha carbon atom next to the carboxylic group. Amino acids are building blocks of proteins. There are 300 amino acids that occur in nature. Among these 20 known standard amino acid commonly occurs in proteins, right? Amino acids commonly occurs in proteins. Amino acid difference from nature. The reach of carbon atom determines the properties. And general formula. These are amino acids. Amino acid structural units of in the proteins. Polymer, peptides, longer chain called as polypeptides are proteins. Polymerizes near liner and unbranched. Each amino acid within the chain attached to two neo-burning amino acids. The process of making proteins is called translation. The process of making proteins is called translation. Involves this step-by-step addition of amino acids to growing proteins chain ribosome. That is called as ribosomes. Growing protein chain by ribosome, the order which amino acids and added through the genetic code. Amino acids are added read through genetic code from mRNA. mRNA means messenger RNA, which is the RNA copy one of the organism of genes. And one of the organism of the genes. Amino acids have travel means glycine and as well as the travel names usually affected by the glycerin. The first obtained by the proteins formed by carboxylic group of one amino acid to the amino group of another amino acid. The bond formed between two amino acids by the elimination of water molecules is called peptide linkage bond. Peptides are further designed as to the di, tri, tetrapeptides, dipeptides, tripeptides, tetrapeptides. Two or four or five amino acids. These are and the classification of polarity basis. Non-polar amino acids, polar amino acids, polar amino acids with no change, polar amino acids with positive charge, polar amino acids with negative charge. Non-polar amino acids have equal number of amino and carboxyl groups. The natural, these amino acids are hydrophobic, have no charge. Polar amino acids is positive charge. Acidic and basic. Acidic and basic amino groups. Right. All of you once again, thank you to everyone. Right. Tomorrow onwards, next intimate to next further classes. Next
Monday, sir.
Hello.
Monday, na sir.
Monday. Tomorrow I will send information.
Okay, sir. Okay,
Particular
sir.
groups lo meeku intimate chestanu. Monday or Monday or Tuesday. నేను చెప్తా I will intimate. Okay na? మీరు
Okay,
separate
sir.
group ayira. Please save my number 9000 11 2991 9000 11 2991.
Okay, sir.
Okay na?
Okay.
I will
Thank you, sir.
inform definitely next class timings and group లో మెన్షన్ చేస్తాము. అప్పుడు మళ్ళీ ఇంటరాక్ట్ కావచ్చు మీరు. ఓకేనా? థ్యాంక్ యూ టు
Okay, sir.
ఎవ్రీవన్.
Thank you, sir.
రైట్.
Thank you, sir.
ఇంకా లోకల్ వాళ్ళేనా? ఎక్కడ? అంజలి, లక్ష్మి, శివకుమార్, భాను, వెంకటనారాయణ రెడ్డి.
Shivakumar, I am from Andhra Pradesh, sir, Kurnool district.
I am also Andhra Pradesh, sir.
I'm also Andhra Pradesh, sir.
Okay, everyone, thank you. Right,
Thank you, sir.
close the
Thank you,
English session.
sir.
Thank you, sir.
Right, close the English session.
Leave all, sir.